So what Hershey and Chase did is that they used something called bacteriophage T2. And a bacteriophage, or a phage, is simply a virus that harms and infects bacterial cells. And in this case, bacteriophage T2, it's a virus that attacks and infects E. coli bacteria in particular. And bacteriophage T2 only contains DNA and proteins. It does not have RNA. So here's the main structure of a bacteriophage. All of this right here, all of this, that's proteins. And this hexagonally shaped head is also known as the capsid head, which is once again composed of proteins, and the DNA is packed and stored within this capsid head. Okay? So what a bacteriophage can do is it can latch onto a host bacterial cell and it can inject its genes into a cell, and then even if the bacteriophage gets removed from the surface of the host bacterial cell, the genes it injects, well, these genes are still present, right? So these genes can keep on replicating, and in the process, they form phage progeny, and these phage progeny keep replicating, so as a result, you get more and more bacteriophage that keeps on infecting the bacterial cells even more, and so on. And eventually, the bacterial cells can burst apart, they lyse open, and then the progeny, the new bacteriophage, come out, and then they can infect other bacterial cells as well. We know that bacteriophage, so far, it injects genes into the bacteria. The question is, what contains the genes? Does DNA contain the genes? Or do proteins contain the genes? Is DNA the genetic material that has genes, or are proteins the genetic material that has genes? Because, for example, if DNA is the genetic material, then DNA should be able to get inside the bacterial cell, first of all, and as a result, once it gets inside, it should have the genes that allows for all this to occur, for the phage progeny to come into existence and so on. But if proteins are in fact the genetic material, then first and foremost, proteins should be able to get into the bacterial cell in significant amounts, and as a result, if proteins are the genetic material, then they should contain the genes that are necessary for the gene replication and the formation of phage progeny, and then cell death, bacterial cell death, for all of that to occur. Okay? So what Hershey and Chase did is that they divided their main tasks into two experiments. So let's analyze experiment number 1. First of all, they had their bacteriophage T2, and then they radioactively labeled its protein coat with an isotope of sulfur called 35S. And the reason why they did this is that here we're focusing on proteins. So proteins and then their smaller subunits, or more fundamental subunits, amino acids, they contain sulfur, but guess what? Nucleic acids such as DNA do not. So they were saying, hey, just to make sure that we're not testing DNA accidentally, let's use something that's only found in proteins and not in DNA. And in this case, 35S is found in proteins, but it is not found in DNA. Okay? And the purpose of radioactively labeling, or radio-labeling something, is to be able to track it later on. So we're going to get to that in a little bit, but let's continue with the process. So once you radioactively label the proteins in the bacteriophage, the phage is allowed to attach onto the host E. coli cells, and it injects its genes, just like I explained previously, and then you can use a blender to vigorously remove the phage from the surface of the host bacterial cell. Now, all of this happens within a solution in a test tube, alright? So, actually, in order to perform the radioactively labeling, tracking, and detection of where the proteins are located, after these three main steps are done, you still have it in your test tube, right? And then you perform something called centrifugation. So you spin it around. So the lightest materials are on the top, and then the heaviest materials are on the bottom. So basically, once you spin the solution around, you track if there's a lot of radio-label protein, or like in the next experiment, DNA, and then you also test the lightmost material and lightmost layers. All right. But basically, what Hershey and Chase found out with the centrifugation process is that the infected E. coli bacterial cell has very little 35S labeled protein, but there is also a lot of unlabeled DNA. So in terms of just having barely any 35S labeled protein, what does this imply? Well, it shows you that proteins weren't even able to get into the bacterial cell for the most part. So if proteins can't even get inside the host bacterial cell, but genes are supposed to get injected into the host bacterial cell, As a result, proteins cannot be the genetic material, right? Because in order for proteins to be the genetic material, one of the most fundamental and major requisites is that proteins, first and foremost, should be able to get inside the host bacterial cells in the first place. Just like genes are injected into the host bacterial cells. But if you find a lot of 35S labeled proteins outside of the bacterial cell instead of inside, then guess what? The genes still got inside. So that means that proteins weren't really even necessary for the genes to get injected in the first place. So as a result, proteins can contain the genes because there's a lot of proteins outside, but all the genes are inside. So therefore, the proteins couldn't be the genetic material that contains the genes. Okay? And then how do we show this within the test tube? Basically speaking, all of the labeled protein is in the lighter layers, and then there's barely any 35S labeled protein in the bottom layers when you do the radio labeling tests. Okay? So next, they went on to experiment 2. In this case, they used an isotope of phosphorus called 32P to radio label DNA. And why did they use phosphorus? Well, it's because it's found in DNA, but it's not found in proteins and in amino acids. Okay, so then the phage still attaches onto the host bacterial cell. You can use a blender to remove the phage. And in this case, with centrifugation analysis, Hershey and Chase found out that the bacterial cell actually has a lot of 32P labeled DNA inside of it. So what does this show? Well, guess what? DNA was actually able to get inside the host bacterial cell by a significant amount. So since DNA succeeded in getting inside the host bacterial cell, well, guess what? Let's take a look at this. Okay, so we know that genes get injected into the host bacterial cell. We've already ruled out proteins, and bacteriophage T2 doesn't have RNA, so it only leaves us with DNA. So if DNA is able to get inside the host bacterial cells, then DNA should most likely contain the genes that allow for the formation of phage progeny to occur. Okay, so here is just a restatement of what I said. So since the DNA from the phage, from bacteriophage T2, gets into the cells by a much higher amount and percentage compared to the amount of proteins that manage to get inside the host bacterial cells, therefore DNA should be the genetic material that contains the genes that allows for the formation of phage progeny to occur.